So it's nice to go from snow to bright sunlight and vitamin D. And 2 points before I start. One, there's absolutely no theory in my talk. This is the motivation, and in fact, I volunteered and I actually wrote to Kavita and Deepa, because I thought that ancient DNA as a field is something that's growing, it's new, and I thought that since we're bringing this in a big way in India, which I'll talk about towards the end of my presentation, what we're doing in India with this tool, I thought as students of PopGen, whether you're doing theory or empirical work, it's important for you to know what are the new sort of emerging subfields within PopGen broadly. So it's going to be really a catch-up for you guys to tell you what the new and upcoming research within this field is. And the second point is that I know that I stand between you and coffee or tea. So if I run over, feel free to tackle me down or tell me to shut up or throw something at me, because I do tend to ramble on when I get very excited. But anyway, cool. Okay, so before I start, can I get a show of hands who's heard of ancient DNA, who's read papers or who's attended talks on this, who's familiar? Wow, yeah, that's more than I expected, so great. I mean, I could just pack up and go. So great, I mean, I don't know what you've heard about it, but hopefully I can catch you up on what's been going on. So really perspectives from the past is what I'm here to deliver. And I'm gonna structure my talk in 3 components. I'll start off by just giving you, again, a little sort of behind the scenes what ancient DNA is. I'm not gonna get very technical because there's limited time, but if anyone's interested in sort of the black box in the lab, what goes on, how we do things, where we get samples from, you can catch me during the coffee break. And then I'll spend some time just to sort of unravel a case study where ancient DNA was used, and particularly the Americas, because it was a large part of my research career. I did my PhD and postdoc on the Americas and peopling of the Americas using both ancient and modern genomics. And then just sort of a couple of minutes at the end to tell you about what we're doing with this tool in India and some other under sort of represented countries or regions of the world where this tool hasn't really taken off so far. So what is the motivation and what really got me and sort of similar, researchers doing similar work is, and just a caveat because I am in the Department of Human Genetics in Chicago, so a lot of my themes will be human in nature, but this by no means, means that ancient DNA hasn't been used with other species as well. So just sort of a general, what we're doing here, what really motivates us geneticists or population geneticists is really understand what drives diversity, right? You want to kind of unravel the various processes that sort of form this huge diversity we see, whether in humans or whether in different species. But again, as evolutionary biologists, particularly working with ancient DNA, what's really fascinating for us is to really catch what's going on, what has gone on in the past, because obviously these evolutionary processes are a product of time. And just catching the diversity today may not be sufficient to tell you or help you accurately model what's built that model over time. And so really what has driven this diversity over time and to kind of catch that in real time. So again, as an example, I'm going to keep going back to humans, so just bear with me on that. But to sort of build up to why ancient DNA really has been helpful and has really helped us fill in gaps that modern genomics necessarily hasn't. So again, coming back to humans, this was sort of the model of human demography back in 2010. This is from a review. And this was based on some early work, primarily with microsats, mitochondrial DNA, Y chromosome, uniparental markers, all modern genomics back then. And this idea, and the sort of 2 main features of human demography were that when there's this very profound bottleneck that happens between 60 to 100,000 years, which is sort of the event that brings our ancestors out of Africa, and that's what you see here. So in Africa, you see these very, very deep lineages, again, corresponding to the ancestry or the ancestral pool of our species. And then about that time that I mentioned, you get this sort of migration, a subset of our ancestors come out and bring with them partial diversity. And then eventually you start getting peopling of the rest of the continents. And so there was a sort of almost a linear view based on modern genomics, sort of how our species went and basically peopled the entire world. So again, this concept sort of came out of this idea that as humans moved out of Africa and sort of sequentially peopled the continents, you have this concept called serial founder effect, which basically means that you're only bringing partial diversity every time you move and colonize a new place. So it's your classic founder effect, but it's serial because you're consequently peopling new places. And this was beautifully shown by multiple people. And this is sort of a graphical representation of one of those results where as you move away from Africa, your levels of heterozygosity sort of go down. So you're dealing with very diverse African populations. By the time you come to the Americas, which is one of the more recent regions of the world to be peopled, you get a sort of lower diversity in contrast to African and Asian populations. Again, showing this sort of serial founder effect and supporting the out-of-Africa migration. But then you fast forward, and this is a review that is, I'd like to say recent, but this is already out of date. So this was published by Rasmus Nielsen and group in 2016, which sort of integrated information since that model that I showed you in 2010 up until 2016. And this sort of brought in various new modern genomic data sets that were published since, but also this crucial sort of missing factor, which was ancient genomics. And so based on that, there was this whole idea that there's a whole different level of contribution to modern humans in the form of these archaic species that all of us have heard, the Neanderthals, Denisovans, even unknown lineages contributing potentially to modern humans. And particularly within humans as well, within human lineages in different parts of the world, there was sort of beginning of an appreciation for the complexity of the process of peopling and evolution of humans since the out-of-Africa event. And this came in the form of large-scale migrations, or the ability to detect humans migrating sort of on a continental level, but also within continental, so within sort of regional scale. And an idea or an appreciation of ancient or past admixture events that, again, contribute to modern cosmopolitan populations. And of course, a little bit of an insight into the process of adaptation in multiple populations and how that panned out over time. So again, adaptations, we're talking about, and we heard some of these concepts already, whether it's lactose tolerance or you have, I lost, so this is supposed to be high altitude adaptation, so some populations who can live in hypoxic environments, high fatty diets like the Inuit populations in Greenland who can really subsist on these high fat diets with relatively low cardiometabolic disorders, which is really quite striking. On the other end, you can look at any sort of diseases, and you start sort of wondering, and there is a pervasive popular sort of concept in sort of the literature that whether some of these so-called diseases or maladaptations may potentially have been variations in the past that may have been neutral or potentially even positively impacting our fitness, but over time, as environments change, as our lifestyles change, whether they sort of backtrack and become these sort of ghastly diseases. And of course, that's fascinating. That's a very interesting take on it. But how do you test that, right, without data from the past? So coming to this idea that obviously, you have genetic diversity across space, and one of the key sort of you hear about this today in modern genomics, human genomics particularly, where everyone's sort of harping on diversity in modern genomics because focusing on one part of the world is just not enough and doesn't capture the diversity. So in the same sort of vein of thought, there's also a concept of capturing this diversity over time. And as you have changes both in terms of demography, whether it's migrations, whether it's admixture, whether it's introgression from different species, or changes in culture or lifestyle, such as changes in diet over time, or diseases over time, epidemics, you have these sort of pressures or selection pressures over time, changing environments over time. And one very quick sort of example to situate you in this thinking is this idea that all our ancestors obviously were hunter-gatherers still about 10,000 years or 12,000 years ago. And then you get sort of between about 5 to 12,000 years ago, depending on where in the world you are, most of us, or most of our ancestors became agriculturalists. There's still some populations in parts of Africa and Asia who still retain the ancestral hunting and gathering way of life, but most of us are obviously eating processed food, westernized diets, and so on and so forth. But this process, as I said, happens in this very crucial time period, which the archaeologists call the Neolithic package. And what this Neolithic package is basically our ancestors figuring out how to domesticate plants and animals, right? And so you have agriculture setting in most parts of the world. And with that, you have this drastic shift in our diet. So you have this pressure on our biological system to change subsistence strategies in quite a radical way. And then, as I mentioned, you have domestication not just of plants, not just agriculture, but also animals. So you suddenly have humans living in close proximity to animals. And again, there's this sort of popular theory about potential for zoonosis when you start having these close contacts between animals or different kinds of fauna and humans. And then talking about changing diets, humans also figure out this very nutritious source of food, milk, and start drinking and digesting it. But as you all may have known or may have heard, not all humans are able to digest milk today. And in fact, some human populations are able to, are the only adult mammals who are able to do so. So no other mammalian species can actually digest milk. And so what brings that about? And then, as I talked about before, there's this time period is also characterized by huge migrations. And these are not migrations that are the original out of Africa, but even after humans supposedly settled in different continents, they're still migrating back and forth. And we can detect this not just in the archaeological record, but in genetic record, because we find signals of admixture between different regions of the world in the past almost 10,000 years. And then finally, this concept of urbanization, again, as humans start settling down as agriculturalists, as pastoralists, they start sort of figuring out we can all live together, cooperative societies, and again, potentially sort of provides a platform for epidemics when you have humans living in large, population sizes. And so it's just to give you a taste of how changing cultures of lifestyles have impacts beyond just the immediate, cultural manifestation in the archaeological records, and how we can, in fact, start to detect all of these in today's biological records as well. So just sort of to finish off this section, in sort of reconstructing models of demography, whether, again, I've been harping on about humans, but whatever species of interest to you. The idea is to capture these processes to be able to sort of, come up with a best fit model of how we got where we got. But in doing so, we're also potentially not just missing out nuances of the past in making this model more accurate, but of course, we may also be missing out on lineages that have gone extinct that may not be represented today. And some of these lineages, in fact, may have contributed to modern day diversity. So again, this impetus to sort of go back in time and capture what's there in the past. So, well, that brings me to the blast from the past. And so this is a tool that we've been integrating, and it's becoming hugely popular, although it still remains quite, let's say, niche, because ancient DNA requires quite a lot of facilities that modern genomics usually tend not to. So for instance, one of the big things that ancient DNA needs is a forensic style isolated facility that not many places have or don't have as yet. And it is quite tricky because, as this cartoon shows you, these are molecules that have been sort of lying in dirt for tens of thousands of years or even hundreds of years, depending on the preservation conditions. So once you extract this DNA, it's not going to be intact. It's not going to be hundreds and thousands of kilobases as it would be if we were to extract DNA from our blood or even hair or saliva. These are highly fragmented. They're highly damaged. So there are mutations which are artifactual, and if you don't account for them, you could potentially skew downstream inferences. But the nice thing is that the last 10 years of doing this kind of research, particularly as genomics sort of crept into the biological sciences, we've been able to garner a lot of data sets, and now we have the ability to start looking for patterns in our data, be it what kind of damage is pervasive in ancient DNA data sets, how do we mitigate that? And then we talk about contamination as well, particularly when you're working with ancient human samples. You, as a human, obviously tend to be your sample's worst enemy, because not just the archaeologist in the field, but also you in the lab, if you're not careful enough, could be contaminating your samples. And because these are damaged, obviously they are low copy number, and as contaminants from modern sources creep in, that could actually flood the signal sometimes, which again will cause you to infer very incorrect things about your samples and past populations. And so this idea that we've sort of really gone up in the last almost less than 10 years. So the first ancient genome was published in 2010, which was, there was one modern genome, 4,000-year-old Greenlandic individual, and the first draft genome of the Neanderthal was published in the same year. And those were just two, so you see like tiny blips there. And then you fast forward to 2019, almost the last 2 years, and now we're missing already the first month of this year. We've already had some publications, so it continues. And this is all thanks to two things. One is the next generation sequencing creeping in, but also as people in the lab have figured out what are the best sources of DNA. So not all tissues, for instance, not all skeletal materials or elements are going to preserve DNA equally over time. So now we have a better idea that there are certain sources in the skeleton that are better able to preserve DNA over time, and we know to target that when we go and sample them. So let's see. In the next 7 or 8 minutes, I'm going to quickly breeze through some popular sort of, let's say, applications of ancient DNA before I sort of lock into the human population history. Because again, for those of you who may be interested in varied applications or varied species, or even varied questions that people are asking with this tool, this will probably give you a taste of what people are doing across the world with this tool. If you want to talk very specifically about any of these, please come and find me during the coffee break. These are in random order, by no means my favorite application or anything. So one of the things that ancient DNA has been used for in the past is to reconstruct past environments, whether it's past flora, past fauna. This can be normally done if you have access to, say, skeletal material from ancient animals, or let's say ancient pollen if you're looking at plants. But one really neat way of doing this is also sampling basically sediment or ice cores. And essentially you have free DNA, and it's like a time capsule, so you basically go back in time, right? So if you're sampling this, the sort of top part closest surface will be the youngest material, and as you go back or down the core, that you'll go back in time. So people have leveraged this, for instance, to reconstruct the past of the paleo environment in Greenland. A lot of this work has obviously been done in the Arctic because it's cold, it preserves well. So this was one of the early papers that came out that introduced this sort of subfield within ancient DNA called sedimentary ancient DNA, where they went and sampled basically ice cores across different parts of Greenland and tried to reconstruct back in time what kind of animal and plant assemblages you would get. And what did that mean for how Greenland was back in time? And what they found was this time between about 500 to 100,000 years, a million years, Greenland was a forested community, so it was very unlike what it is today, which is complete tundra covered with ice sheets. So again, it tells you something about climate change over time, about what sort of places may look like. If you look at it today, obviously it has a very different story to tell, right? And also to a quick fact, this DNA, or this study basically touched the max limit of what is recoverable in terms of the temporal scale of DNA. So the sort of theoretical limit to what we can extract is about 1 million years, and this between about 800,000 to 1 million, this study sort of touched that limit already. Right, and just to sort of another quick interesting example where they use these sediment cores. So this one was ice core, and I told you this was up in Greenland because preservation is great, but also sort of in the temperate area. there was a study that looked creatively at a means to reconstruct where and the route that people moved, right? So one of the very contentious questions in the peopling of the Americas is which route humans took to enter and people the Americas. And the reason for this debate is because until about 12,000 years ago, North America was covered by large ice sheets. It was still Ice Age, the last Ice Age. And so the entire Canada and northern North America was completely shut off, right? So people could probably come up till here, but then not enter down south. Yet we do find archaeological sites and presence of humans down in southern US and South America, obviously. And so the question is that how did people get here and when did they come down? When did the ice sheets start melting? So from climatology records, we know that there are two routes that open up over time as the climate starts warming again. So the first route that opens is the Pacific coastal route, which opens up about 16,000 years ago. And then there's an interior ice-free corridor, which basically tracks the modern Rockies. That opens up about 14,000 years ago. And so the question here was, well, how did people move? And the premise was that as people move, you would leave sort of traces of DNA behind. And can we actually trace this? And so the researchers basically looked at this and they said, well, we would have to basically sample sediments from this and be able to then compare and see whether we could actually detect remains or some sort of traces of human activity, right? Unfortunately, this route is because since 12,000 years ago, the ice sheets have melted and the ocean levels have come up. So that route is now underwater. And so you would have to do marine archaeology to be able to retrieve that sediment out, and that's a lot of work. So basically, the only route that's accessible is this place in the Rockies. And so they went up to a few lakes up in the high altitude, and they sort of dug in and got cores from the lakes. And what they did was, just gonna go to the results. So what they did was they basically looked for presence of biological viability, because the rationale was, so this is almost, this whole route from north to south would be about almost 1,500 kilometers. That's a long way to go if you don't have food as humans. And so once the ice starts melting about 12,000 years ago, the soil is still, it's not rich enough. So it's gonna take some time to regenerate, then for plants to start growing. And then once plants start growing, that's gonna attract the animals in, and then that's gonna bring your hunter-gatherer ancestors in, right? Because they're gonna follow their prey into and through the route. And so what they see in their sort of records over time is they start seeing biological viability in the form of both pollen, so plant remains, as well as traces of fauna, different animals, about 12.5 thousand years ago. Before that, they get really nothing. So that's, again, that tells you that the soil is still not rich enough to regenerate. So that's sort of your minimum time period, or that sort of seals the time when humans could have used that interior route. So that's about 12,000 years. But in the archaeological records, you find some of the old sites about 15,000 years deep down in south and southern Chile. And so obviously humans needed to have come down before 12,000 years ago to get down here. And so by default or by exclusion, they came to the conclusion that they would have had to use first the Pacific coast to come down, and then later they would have had the option to use this, but initially, or the early peopling would have happened along the coast. Right, okay. So another application has been to look directly at animal or fossil remains from animals, and one of the big attempts at using this has been to resolve this question of megafaunal extinctions. So megafauna are defined as animals that are above 45 kilos. A lot of them in North America, or actually northern Eurasia, have gone extinct, and this extinction happened about 10,000 years ago when there was a drastic, again, shift in the climate patterns. So species like the muskox, the woolly mammoths, woolly rhinos, and big sort of equids that were living in these areas went extinct. And the question was, and there were two sort of schools of thought. One thought that it was actually humans, because again, this was the time when humans were entering the Americas, and whether they had overhunted and killed these off to extinction. And another competing theory was whether it was climate change. So again, using DNA evidence or DNA retrieved from fossils from these different species and looking at basically the population size changes and whether that corresponds to the evidence from archaeology of humans entering versus climatology evidence of change in climate. These researchers were able to come up with sort of models of which species might have succumbed to humans versus climate change. Another application has been obviously the detection of archaic species. This is something that's really made big news because one of the inferences has been that not only were these archaics existing at the time when humans came out of Africa, but they actually also interbred, there were genes or genetic material that introgressed not just from them into our ancestors, but from us into them. So there's a lot of back and forth. There's various species that have been since defined, and also there are some unknown species, like I said, that still don't have any fossil evidence, so they're ghost species. And different papers now started not just looking at the identity or demography or the contribution of these species, but also looking at what are the functional consequences of these introgressed regions in modern human genomes. So again, because of time, I'm not going to go into these. I just want to give you very briefly kind of themes that people have done. And I will probably just talk about this and skip because there's limited time. So just the last example I want to give you is looking at selection. As I mentioned, there are ways to do this when you have enough ancient material. Now, this is not possible in many parts of the world because you need, obviously, population size data sets, which there's only one region of the world where we have leveraged ancient DNA data sets to get population-wide data sets, and that's Europe. And so there were two papers. This is just a review paper that's actually summarized their results. But there were two papers that came out of one in Denmark and one in Harvard in 2015 that published 100 individuals all the way from the last 10,000 years to more sort of historical times from across Europe. So all different regions. And essentially what that gave us a window into is not just how the demography of Europe has sort of changed over time and how that's contributed to the modern gene pool, but also to look at sort of interesting adaptations or interesting variations that we know of today and see when those arose back in time and what may have actually caused them to rise in frequency. So one classic example is lactase persistence. So again, going back to this idea that only some human populations in the world can drink and digest milk. And some of the sort of highest frequencies of this particular adaptation are seen in Northern Europe. So Scandinavians have some of the highest frequencies of being able to drink and digest milk, so that particular mutation in the lactase gene. And so before these papers were published, the sort of popular thought was that this happened, the fixation of these allele in these populations happened about 10,000 years ago when humans domesticated, they adopted agriculture and settled down, because obviously, the thought was if you domesticated animals, you would have figured out that you could drink milk. But when you looked at the data here, so this is each dot is an ancient sample, and the crosses as you go further in time to present time, these are all modern populations. And the color codes are basically corresponding to different countries they come from. So as you see, you go from sort of southern parts all the way to the north. So this represents the Scandinavian countries. It's pretty much you're getting near fixation of this particular adaptation in the north. But when you look at the ancient samples, which is all these circles, over time, the rise in frequency is only really significant about 5,000 years. So it's not 10,000 years when domestication actually happens, contrary to what people have thought. And when you actually go back to the archaeological records and see what happens about 5,000 years ago, what happens is there's a huge migration into Europe from the steppes, which is this area. So there's a huge population migration that comes in. You see this in the genetic record as well, because suddenly the resident farmer populations that we have in the ancient context, they're sort of, it's not a complete replacement, but you see an inundation of their genetic profile with this new genetic input that comes in from this ancient population. And this ancient culture is actually a pastoralist culture. So they've been drinking milk for we don't know how long, but they have there is evidence that they have been milking different fauna, and they come in and they bring that adaptation with them. The resident farmers do not have that in Europe. So when this migration happens, when the admixture happens, that particular variation gets transferred to the resident Europeans, who are now basically a mix of what they were with this new population that comes in. And that gives them a definite advantage, particularly as you're a farmer in times of drought when you may not have access to food. Milk is something that can still sustain you as long as you have healthy animals tied to your backyard, right? And so you start seeing this rise in frequency really when that big migration happens. And so this, again, gives you the power of going back in time and revising concepts, not just in genetics, but also in the archaeological records. Okay, let's skip, because is it almost coffee time? Right. I feel like I haven't even got to the crux of my talk. But really the biggest application of ancient DNA has been to study ourselves. So in reconstructing population histories, and so very quickly I'm going to breeze through the Americas and what we did with an ancient sample. So again, Americas, although they've been one of the last big continents to be peopled by humans, so the earliest evidence about 15,000 years ago of humans, there's still a lot of unanswered questions in archaeology and genetics as to who the source populations are of the modern indigenous populations and Native American populations. When does it happen? How does it happen? This we saw, I gave you a little taste of that through the sediment analysis. And what happens within the Americas? How do they diversify? Because today there's immense cultural, phenotypic, genetic diversity all the way from the Arctic to the southern tip of South America. How does this happen within the Americas? So all these are open questions that are still being addressed. Because I won't have time, I'm just going to go into question one and how we sort of address the question of who the founding population was using ancient DNA. So there are three competing theories. One is that the source population for all the diversity and when I say diversity, this is the indigenous populations, not the post-Columbian European contact. And the prevailing theory among geneticists and anthropologists and archaeologists is that the founding population comes from East Asia. And let's just skip all of this because we don't have time. And this has been supported by early work on mitochondrial and Y chromosome work. And then the second competing theory was that they come actually because some of the early sites are in South America. Some archaeologists claim that they come from Polynesian islands into the south and then move up. And the third hypothesis, which is really the most controversial, is that they come from Europe. And this is based on some archaeological similarities in stone tools, so nothing to do with genetics. And of course, this touches upon the sensitivities of indigenous populations because of the post-Columbian contact or colonization from Europe. But really within genetics, what Native American populations have been depicted, are essentially an offshoot of East Asian because that's really what the mitochondrial early work on microsatellites told us. And that was up until sort of 2010 or 2014. And what changed it was this DNA evidence from one single individual. This was a little boy who was buried about 24,000 years ago in a site called Malta, which is in Siberia along the Lake Baikal. And we got access to a tiny bit of bone material from this child, which is stored in Russia in one of the museums. And this is a reconstruction of the child's burial. And we were able to get DNA. We were able to get, in fact, quite a decent amount of DNA to almost get about 1x coverage of his genome. I know for modern geneticists, 1x is nothing, but for ancient DNA, that's a lot, trust me. And we can still do enough, particularly things like mitochondria because they occur in huge copy numbers. So even at 1x average whole genome depth, you're able to get, on this sample particularly, we got almost about 75 to 80x on the mitochondria. What the mitochondria told us is this is a frequency of where modern signatures of that ancient boy signature occurs. And you can see that he was found here, but really his mitochondrial lineage, so his maternal lineage, resembles Western Eurasia more, modern Western Eurasians. Similarly, because he was a boy, we were able to look at his Y chromosome signals. And again, it doesn't resemble populations who sort of live in this area much, but really he's closer to populations who live in Europe and Central and South Asia more. And when we looked at, and this is sort of a cartoonist version of our PCA, but when we sort of compared the autosomal genetic signals and we plotted basically genotype data from as many populations we had access to data from at that time, and so they're color-coded according to different regions of the world, and we overlaid our ancient genome on top of that. What we found were two striking observations. One is that he is not really related to modern-day Siberians. And second is that falls on the PC1 on a client that's defined on one end by, again, these Western Eurasians, and on the other end by Native Americans. And this was sort of signals that we really did not expect at that time. So it took us a little bit more prodding and modeling to come up with what was going on. So again, in the interest of time, I'm going to skip all the analyses, I'm sorry, but really want to present the model to you. But this was the first time that using ancient DNA, we were able to present a revised dual origin model for Native American populations, who traditionally, like I said, were actually only seen as an offshoot of East Asian diversity. But what we find is about 60% of the ancestry of modern indigenous populations in the Americas comes from an ancestral source that is related to modern East Asians. But up to about a third of their genome is actually contributed by an ancient lineage, which is defined by that child that we sequenced. And that 1/3 is quite a substantial amount. Sometimes it's actually seen potentially as regions in indigenous population genomes, which may be potentially European post-contact. But in fact, we now know that some of those regions are not just due to the modern recent colonization gene flow, but actually a little bit more ancient, almost as ancient as 24,000 years old. So let's just skip all of that, and let me just tell you in 2 minutes, before the coffee gets cold, what we're doing in India. We don't have any results because I've actually just started my group in Chicago. We're very new, but just to give you a sense of the kinds of projects we're establishing in South Asia. So to kind of give you a primer for that, this is a map of all the data that was basically all data that comes out of ancient samples. Back in 2018, this was a review that summarized it. Each red dot represents a genome or a genome-wide data set from an ancient individual. And as you can see, most of it clusters in Europe and Central Asia. This is revised since 2018. We do now have some genomes from Africa, which were actually just published last year, some more from the Americas, but it's still, the disparity is very evident, and it mirrors almost the sort of differences you see in modern genomics as well, where you have most genomes coming from peoples of European ancestry. And so motivated by this, my group wanted to sort of work in areas where sort of traditionally this kind of work has been underrepresented, so we can bring up or raise the diversity in ancient genomic data sets as well. So that's currently my group. We spent quite a lot of time, almost our first year, coming up with a catchy name, because apparently brand is everything. So we're a Genscape lab, and we study contemporary and ancient individuals. But anyway, moving on. So research team 1 that we've started now in collaboration with archaeologists, as well as modern communities across India. We've expanded now to Sri Lanka and Pakistan as well, collecting ancient samples and really piecing together the over time, again, like we did in the Americas, the formation of the gene pool of modern-day South Asians. And you may have heard some of this already in the news. This is the site of Rakhigarhi. About a couple of months ago, there were 2 big papers out of David Reich's lab in Harvard in collaboration with Indian researchers, where the first ancient human genome from South Asia, from the state of Haryana, so that's the site, came out. It was extremely poorly preserved, although morphologically extremely well preserved, but biologically extremely poorly preserved sample. They were able to finally get from, I believe, more than or close to about 1,000 different Illumina sequencing libraries, just about close to about 30,000 SNPs were workable at the end. So it's a very, very scanty data set, but still they were able to push that and leverage as much to understand really the current working model of how South Asian gene pool was formed. Again, I don't have time, but this is all published, so you can go back to this. But this was a single sample from a single site, so we've expanded since, and this is a map showing all the samples we have access to, and we're working in collaboration with a lab in Lucknow, so all the work is being done locally. We are not taking any samples outside India. And as you can see, we've expanded to other parts, and we have some dots now in Pakistan as well. So hopefully this is going to give us a much more comprehensive outlook over time, and these different colors represent different time periods as well. So we covered basically almost the last 8,000 years in our sample set. Now, caveat, this is South Asia, it's extremely hot and humid. So a lot of these samples, they've already failed. So we put in a lot of money for nothing really, but that's really how ancient DNA goes. The preservation is extremely poor, so just like the previous study, for a lot of these samples, we are really scraping the barrel to get workable SNPs to be able to intersect them with the modern data set so we can actually do some PubGen. But still, we're going to keep working on this, and hopefully, particularly, we're focusing on places like the Himalayas because it's high altitudes, it's colder, so we may have access potentially to much better preserved samples. Right, and we're going to skip 2 and 3, but really 2 and 3, if anyone's interested in talking about pathogen evolution, because we're extracting ancient bacterial and potentially viral sequences from ancient humans, so we can actually analyze infections over time. So if anyone's interested, again, come find me. Coffee break's gonna be intense if anyone's interested in talking to me. And the last thing is, we are heavily biased towards humans in our lab, but we're also interested in how humans have impacted on the society, on their immediate biota and environment. So we're also looking at the process of domestication in South Asia. There's a lot of this work that has been done in Europe and Near East, and there have been establishments of independent centers of domestication, particularly in the Near East. But a lot of archaeologists believe firmly that South Asia and China as well were independent domestication centers for a lot of these species. And so that's one of the open-ended questions that we're trying to answer using ancient DNA. And that's all I have for you. Sorry, it was a bit of a breeze, but my motivation was to spread the word, really, and have you guys appreciate that there is this small component that's trying to sort of make its way into the Indian, South Asian context as well. So do watch out for papers from these themes as they keep coming out of my lab. Thank you.